Okay, so let's start with the recording and we can start today's session. So, can tell if my screen is visible to you. Okay, so good evening to the week 9 discussions on the structural biology course. So, today what we will do is that we will be discussing the assignment week 8 assignments and also the solutions of the week 8 assignments. And also, I would like to talk about the cryo-electron microscopy and microscopy as a whole, because if you have looked into Sir's lectures, you will find that he has started with cryo-electron microscopy by giving you a brief introduction about the microscopies, different microscopies, and how those microscopies are different from cryo-electron microscopy. So that Sir has discussed already with you. Okay, so I will like to give you the summary of those of that week's content, and that will also help to solve the week 8 assignments. So first, let me give you a brief summary of the microscopy part, and then we will discuss the week 8 assignments. As well as I will also be giving you some practice questions in my slides so that you can practice. If we have time, we can practice here as well. And if the time does not permit, we can what you can do is that you will have these slides, so you can see those questions and try to solve those questions, because the questions that I have collected, that is from previous years assignments and your exams, so it will help you in the exams if similar kind of questions are asked. Okay, so before going to the today's week's discussion, if you have any doubt, you can ask me, since only one of you is there. So Shani, you have any doubt from the last class discussions? Yeah, in week 8 assignment. Okay, so can you hold that for a few means hours? Means we can discuss the assignment at the end of the session. Okay, okay. So other than that, if you have any doubt, means from previous discussions. Okay, okay. So, means if you don't have any doubt, so let me discuss with you before starting today's session. Let me discuss with you little bit about two kind of spectroscopies that I didn't discuss in the last class. As if, hello, hello, yes, my voice is breaking. 1 minute, 1 minute. I will solve that. Hello, am I clearly audible now? Okay, okay. So, if you find any means the voice breaking is happening again, you can then ask me. Um, okay, okay. Hello. Then let me start to this session. Okay. So if it is happening again, you can just let me know. So, yeah. So I was telling that last year a student asked about two spectroscopies which are not discussed by sir, but I just wanted to give you a brief knowledge about that. So these are two new topics, but yeah, in brief I can tell you. So one is electron paramagnetic resonance (EPR) spectroscopy. Another one was neutron scattering. So that was not discussed by sir. that is just for your extra knowledge, since you don't have any questions, so that's why I am discussing a little bit extra. But this will, I don't think this will be asked by sir in his questions you will get or assignments you will get, but this is just for your knowledge. You can read, I will share some contents about this, you can read, go through them, and if you have any doubt you can please ask me. So the first one is electron paramagnetic resonance spectroscopy. So if you remember, I said you that nuclear magnetic resonance NMR is a kind of spectroscopy that we have already talked about. So this is a kind of, so similar to NMR. So in NMR, if you remember, what we were interested in is that the nuclear spin. So if you remember, the what is the spin of your nuclei, that was important in nuclear magnetic resonance spectroscopy. So similar to that, if someone try to look into the spin of an electron instead of the nucleus of an atom, then you get to this electron paramagnetic resonance spectroscopy. So this I said you in a very simplified term, but the actual thing is that since this name is electron paramagnetic resonance, so there should be some paramagnetic nature, means there should be some odd number of electrons in your atom which are EPR active. Okay, odd number of electrons if present, then they are both paramagnetic in nature. Means if you apply a magnetic field, it will be pointing in one direction. Okay, so they are paramagnetic. So this paramagnetic means paramagnetic types of atoms which have odd electrons. There, if you apply instead in NMR, what we were doing is that we were applying a radio frequency wave that was means that was in the range of the energy difference between the two states of the spin, the parallel spin and the antiparallel spin of the nuclei. Similar to that, the electron spins, the plus of and the minus of spin, they will also have one energy difference in there, because there will be two energy states for these spins as well, electron spins as well. And that difference can be compensated when you use a microwave instead of radio wave frequency, where we use microwave. And once you apply that energy difference, you can get different spectra similar to the nuclear magnetic resonance. Okay, so here we are making use of the electrons instead of the neutrons. Okay, in electron paramagnetic resonance spectra. So for each amount, each different atoms, you will have different spectra because of this difference in the energy levels of their plus of and minus of states in the of each and every atom. So you will get different individual spectra. So that is your, in nutshell, electron paramagnetic resonance. What is neutron scattering? Neutron scattering is when you means if you try to look into that, that when you hit a means gold foil with an electron beam or a beam of alpha particles, then you will find that these neutrons, the means if you remember how the neutrons were get means were being discovered because of this fusion and fusion of these nuclei, you will get these neutrons. Okay, and the just like in here you try to make an analogy with the X-ray crystallography, where you were actually using the X-ray as a source and it was hitting your sample, and once it was hitting your sample, the amount of electrons that it will scatter the X-ray. So the more the amount of electrons, the more is the scattering, and you will get the electron density map in a detector. So instead of your electron, if your neutron starts scattering the source light, and that is actually giving you the means the spectra, then it falls under this neutron scattering in a nutshell. So what is the advantage of neutron scattering over this X-ray crystallography is that in X-ray crystallography, you remember that if you have means similar amount of electrons, for example, for two isotopes which have maybe similar amount of electrons in them, then the problem is that you can't differentiate between those two. by using simple X-rays and um, emitting this number of electrons. But for neutrons, if for those elements there will be a difference in the number of neutrons, then that can actually give you distinct spectra of two entities which have similar number of electrons. Okay, so then this neutron scattering is handy. Okay, so these are means in a nutshell. I have just discussed with you to give you some interest towards these techniques, because these are some new techniques that you can actually go and see. I will also share some materials details about, because there are a lot of mathematics for this EPR as well as this neutron scattering. So I am not discussing those because this is not under this course, but just that for your extra knowledge you can go through them. Okay, so that was what I wanted to discuss. Now let us start. Means spectroscopy we have seen. Now let us go to microscopy, another property of light. Okay, so the main discussion that we will do today is the starting of this cryo-electron microscopy topic. So if you remember, when I talked about the different three-dimensional techniques that we have in our means for actually visualizing your structures in 3D, I mentioned about X-ray crystallography, NMR, and cryo-electron microscopy. So X-ray crystallography and NMR we have discussed. Now we are moving to the third technique, that is the cryo-electron microscopy. So from the name itself, it is giving you some idea what kind of microscopy it is. Okay, so we will look into the basics of microscopy. So what is microscopy? We will define that in the next slide. But before that, just to give you an idea about the cryo-EM. So it is cryogenic electron microscopy. In short, that is cryo-EM, is an electron microscopy technique applied on samples cooled to cryogenic temperatures and embedded in a environment of vitreous water. So what you have seen for the other two techniques, like the X-ray crystallography and NMR, you have seen that for X-ray crystallography you need to form a crystal. Your protein needs to be in a crystal, and then you will actually diffract it and get your electron densities, and from that you will get your three-dimensional image. In NMR, what you got is that your protein is in a solution, and you can get the chemical environment change that your protein or your molecule can is taking place in that media. That changes you can actually visualize with the structure, 3D structure of your proteins. But in both of them, we talked about some disadvantages and advantages. So we talked about the mean size limit of what you can image in both of those techniques. Means in what resolution. If you can talk about in higher resolution, what is the size limit? What are the size limit to which you can go for X-ray crystallography and NMR? For X-ray crystallography, it can be means greater for greater size. The X-ray crystallography you can get the image, but that will be not in a in vivo condition. Means it is in a in crystalline state. Your protein is in a in crystalline state. So the nature of your proteins, what it is behaving, whether if it is interacting with a substrate, if it is an enzyme, those informations you can't get in X-ray crystallography. Okay, and in NMR, though you can get those information because your proteins are is in a solution, so you can get those information, the dynamic changes of your protein with the solution. But the problem with NMR is that you can't go to higher molecular weight or higher molecular size, because if you go above 30 kilodalton, or it will start, the problem will start from above 20 to 15 kilodalton. If you go, the resolution, there is a compromise with the resolution of your image. Okay, so the resolution will be bad, and you will not be able to distinguish between the noise and the of a noise and your NMR peak signals. so that will not be serving your purpose. So then scientists were thinking that what should we do? In both of them, we are getting a part of the information. And in cell, we know that no protein is acting alone. The best example of this is the ribosome. So in ribosome, as you know, that this ribosome is the main machinery for your protein translation. But ribosome is not only made up of protein, it is made up of proteins, RNAs together acting with one another, some enzymes, and they will actually working with one another to for the protein translation to happen. So there was a question that can we now actually image the three-dimensional structure of a ribosome? Because then only we can actually know what is happening inside our cell. Okay, in vivo like condition, can we do that? Can we image the three-dimensional images of these in vivo like conditions in such a way that we get a complete picture, and also we get a means there will be always a compromise with the resolution, but you can actually get the whole process also out of it, what is happening near to in vivo conditions. Then people came up with this idea of this cryo-electron microscopy. Okay, so where you actually freeze, you cryo freeze, more specifically, you cryo freeze your sample so that your samples are embedded in vitreous layers of water. So if you remember what is vitreous means, you in your eye you have this vitreous humor. So what we mean by vitreous means there is a thin, very thin layer of water in which your proteins are embedded. Okay, so that thin layers of water, they are in in such a way that the outer environment of that water is freezed, whereas the inner environments of the water is still like in a liquid form. So it is cryo freezed, but it is in the proteins are in more in a in vivo condition. So that's why this process you will learn about vitrification is done. Means this is very important for a cryo EM to work. So then you should ask a question that why we need to freeze our sample? Why we cannot directly get the image of our sample directly through electron microscopy? Why? What is the role of this cryo freezing? Okay, so these questions should come in your mind. So if you have those questions, just keep it in your mind. You will get the answer. Okay. So this technique was so important that the Nobel Prize was provided to Dubochet, Frank, and Henderson for this discovery of this cryo-electron microscopy in 2017. Okay, so then from what you got is that in the subsequent years you got the first three-dimensional cryo image of bacterial rhodopsin. So that was the first three-dimensional cryo image that was being submitted in PDB. So what is PDB? We will also discuss little bit today. Okay. So PDB is a means database. Those who don't know that where you actually submit your three-dimensional structures. So I talked about it little bit while when we were discussing about X-ray crystallography and NMR. Okay, so uh, and then so this cryo discovery is also, if you see always my front slides, I always give you two images. So this ribosome cryo-electron microscopy image was also means when being captured, and we also got the three-dimensional view of the ribosome and how different parts of the ribosomes are actually acting for this protein translation. So and that is done by Venki Ramakrishnan. So if you remember his name, and there were other two scientists, so Ada Yonath and Tom Stein. Okay, so they three got Nobel Prize for the discovery of this ribosome structure with the help of this cryo-EM. Okay, so that is the importance of cryo-EM. Now, so cryo-EM is a type of microscopy. So I am going from the basics of microscopy to the cryo-EM. Okay, so uh, the properties of the light can be used in two ways. So first, we talked about the quantitative nature, where we are actually more interested in the intrinsic properties of a matter. Means what where we are interested to know what are the different chemical functional groups, or how this intrinsic properties of the matters changes with certain conditions. So there you are actually quantitatively determining something with the help of the nature of this light. For example, the source through which you want to visualize that properties. So that falls under spectroscopy. So that we have talked in our previous session. Now we are more interested in qualitatively determining the image. Means here we are more interested to look into how that entity looks like. So the properties, nature of the properties which we have defined, that falls under this spectroscopy. Whereas in microscopy, you want to actually visualize that whole means the entity as a whole, and you want to find that how these different atoms are organized to make that particular structure or particular entity. So there are different limits to which you can actually divide the different entities. Means your eye is the best example of a kind of microscope. It's not a microscope, but it is like a means it is like a camera which can capture to the beautiful images of the nature. But it also have one limit to which it can. So that is the limit of your resolution. Means it cannot see something which is beyond the resolution of your eye lens. Okay, so the eye lens resolution is still the your finger, your man's height, the hand palm. It can be easily visualized by eye. Now, if you want to go deeper into means in micron level, like the size of your bacteria, so that can be visualized, or the size of your cells, or the thickness of your hair, that can be visualized easily with your naked eye. For that, you make use of certain lenses, or you make use of the microscopes. Normally, these light microscopes are falling under that range, to which you can actually visualize this hair cell and the bacterial cell to the micron level. You can visualize with the help of light microscope. But what if we want to visualize something which is in angstrom or nanometer length, which is much smaller than the bacteria. So remember that why X-rays are used for this X-ray crystallography, because I said that the wavelength of the X-ray falls in a means range of the distance between two atoms in that is around 1.5 angstrom. That's why we were using X-rays. Now, if we are interested to go much deeper into to electronic level or to atomic level, so that cannot be visualized by normal light microscope, because that the wavelength of your visible light does not falls under that small entity, or the small wavelengths that you are interested to measure. Okay, so for that, you need to go shift your gear from normal light microscope, where you use photons. Then you need something which has a wavelength that is smaller than your photons. So then people try to wonder that what is that which has a wavelength much smaller than your photons. So then they found that okay, so there are electrons which can serve our purpose. So they actually they actually started the field of electron microscopy, where you can actually visualize the viruses as a whole. Okay, then various macromolecules, small molecules, which cannot be visualized by normal light microscopes. So in electron microscopes, you have this cryo-electron microscopy. So then, what are the different types of microscopes? So that is divided on the basis of two different bases. What is what interacts with sample, means what are you using for visualizing your sample? Means there should be some source which is interacting with your sample and giving you some properties that you are utilizing, that those properties you are utilizing from the matter to visualize it. So what interacts with your sample? So if it is a light, then it is known as optical microscopes. Means normally optical microscopes, you have the source of your means visualization from where you want to visualize your sample. The source is your light or the photons. Then the electrons, as I discussed, if the source of your visualization is a gray is a bunch of electrons, then it is falling under electron microscopy. Then there can be certain probes which are known as scanning probe microscopes (SPM). So if you remember, there is a scanning probe microscope known as atomic force microscopy, where you get three-dimensional images of your samples. There what it utilizes is that it does not utilizes this electrons as a source or the light as a source. Instead it has a probe which is attached to a cantilever. So there is a laser light which is coming and hitting that cantilever, and this that cantilever is elastic. So that will follow the Hooke's law. Means if you apply certain force on that cantilever, so that probe will go and touch your surface. And on the basis of the unevenness of your surface, it will try to probe the three-dimensional image of your surface with the help of your quantum electron tunneling. Everything is there. So you are basically imaging the position of your cantilever through that laser beam. Okay, so you are actually you are actually imaging your sample with the help of a probe. So that falls under this probe scanning microscope like the AFM, atomic force microscopy. So this is one type of classifications in which you can actually divide your different types of microscope. Another thing is that how you want to visualize it, whether it is point by point or you want to visualize it as a whole. Okay, so if it is like you want to visualize each point at a time, means you are moving from one point to another point and you are trying to detect the pixels of that point. That is known as scanning point or the scanning point microscopes. For example, like confocal optical microscopes, scanning electron microscopes, and scanning probe microscopes fall under this category. So you actually scan each point when you are moving through the sample. Another type of analysis can be done is that you are analyzing the image of the sample as a whole. You are not scanning point by point, but you are looking into the sample as a whole. So the microscopes which fall under it is like wide field optical microscope and transmission electron microscopes. These actually fall under this category. Okay, so what are these different microscopes we will be discussing? So moving into the light microscope, which is the simplest, I would say. So in light microscope, you have two types. One is simple microscope, another one is your compound microscope. Okay. So what is a simple microscope? A simple microscope is a type of microscope that utilizes a lens or a set of lenses to enlarge an object through angular magnification alone. What it means is that you will have your object and you will have a single lens through which your light will be moving, and it will be providing you a means image but a less amplified or less magnified image, and it will be only able to give you a angular amplification, and it will give you a virtual image. As you remember, these virtual images cannot be directly detected under a detector. Okay, so these simple microscopes will not be able to give you the whole nature of the or whole magnification of your sample. It will be giving you only a angular magnification of your sample because it has only single pair of single lens. Okay, but in compound microscope, what you use is that you have different combinations of lenses which is serving different purposes to give you a larger magnified image of your sample and larger magnified real image of your sample, which you can easily detect. Okay, so a compound microscope is a microscope which uses a lens close to the object being viewed to collect light, which focuses a real image of the object inside the microscope. That image is then magnified by a second lens or group of lenses that gives the viewer an enlarged virtual image of the object. Okay, so what it happens is that you will get you will get an amplified, a magnified image of your sample in the compound microscope, because there you are using two lenses. So one is your objective lens, another is your eyepiece lens. So both of them has different levels of magnification. So if you are using here a 10x eyepiece, then it will objective lens will also have some x magnification, and the net magnification that you will get of your image is the multiplication of this magnification power of this eyepiece lens with your objective lens, and you will get a magnified image of your sample. Okay, and you will get a complete image. In simple microscope, it was only your angular image that you were able to get. Okay, now if you remember, I was telling you there are different kinds of lenses that are present here, like your eyepiece lens and the objective lens. Okay, so what is the role of your objective lens? Let us look into that. Okay, so what are the different roles of your objective lens? So the one of the major challenge that you get while in if you are working with a microscope is that there are different kinds of aberrations that can happen. So there can be some spherical aberration as well as there can be some chromatic aberrations. So what are these aberrations? Aberrations means certain defects while getting the image of your sample. So imagine your sample is not evenly distributed, it is uneven. So there is some samples, mostly if you consider at the center of of your dish, you will have your sample, and for example in the periphery there is no, means the density of the sample is less. Now, the way the light will interact with your sample in the center and the light that will interact with the sample at the periphery, that will be different, because the electron density or the density of your sample is different in different regions. So in center you have a larger density, in periphery you have small. So then you will the deflection of the light or any property that you are detecting finally with the detector, that will be different for the periphery and for the center where you have higher density. That will lead to different defects or aberration in your image, that will give you a blurry effect to your image, and that will compromise the resolution of your image. Okay, so to get rid of that, there are different filters and different kinds of lenses, different kinds of objective lenses that one can use to minimize those aberrations. So that I have talked about this spherical aberration. So whatever I have talked now, that is about this spherical aberration, where light rays hitting the periphery will be more refracted than the rays hitting the center of the lens, because the refractive index of your sample will differ on the basis of the density of your sample throughout your big sample area. Okay, so that will differ. So on the basis of that, the refraction of your light falling onto it differs, and you ultimately you will get a aberration known as spherical aberration. Now, what is chromatic aberration? So, if you remember when we discussed about the topic of this, when you are means passing a white light through a prism, so that white light gets splitted into 7 different colors. Okay, so that we mentioned. So now this white light will got splitted into in different colors. So that how it happens? That happens because of this. The matter actually interact with this white light and try to emit different wavelengths of light, and that is dependent upon the type of matter that you are using. So if you are while in chromatic aberrations, what happens is white light of passing through a simple lens, each wavelength will be refracted to a different extent. So when you are passing that white light through a lens, it will be split into 7 different colors or 7 different more specifically we can tell that 7 different wavelengths, and these 7 different wavelengths will actually interact with your samples in different way. Okay, so that will lead to several aberrations or several defects in your image. So that falls in under your chromatic aberrations. And to minimize these aberrations, you have different kinds of filters and the different kinds of lenses, like the different kinds of objective lenses. So what are the different roles of your objective lens? First one is to gather light rays coming from any point of the object. Now what is if your diffraction means these lines behave at different angles, it get refracted from your sample. Because of this unevenness of your sample, you need something which can capture this whole light and again concentrate it in and into one point. Okay, so what it will do is that it will actually gather the light from any point of direction, so that this light can be condensed with the help of different lenses are there, like condenser lenses are there, which will actually condense this light, and that will fall into your object so that you can get a very fine image of your sample. Okay, so this blurriness of your samples is avoided because you are capturing those light and condensing and letting it fall into a sample. Okay, then to unite the light in a point of image. So that brings me what I was explaining, that once it falls into your object, it may again scatter. So it needs to be united to a point of the image. Means each pixel of your image should not be fuzzy. It should be having a means higher resolution. So that can be also given by this objective lens. And finally, to magnify the image means it's something which is very small. If you visualize it directly with the light microscope, you will find that the image which is found, that is very small, and you can't visualize the entity, the different properties of the entity, or how the different structures in that cell are made up of. So that for that you need to magnify that image. Okay, and see in a means in depth, means how it looks like. Okay, so that magnification is also provided by your objective lens. So and also finally to correct these aberrations, so there are different filters along with the objective lens that correct this kind of aberrations that we discussed. Now this is a table just for your means knowledge. Means if someone so there can be some doubt, like if someone asks about the magnification of a of a microscope, then if someone tells that only since the eyepiece magnification is 10x, so you will get the whole magnification of that microscope is 10x, that is wrong, because it depends upon your objective lens as well. So if you are using a 4x objective lens, then the total magnification of your image will be 10 multiplied by 4, that is 40. Now if you use 100x objective magnification, then it will be the overall magnification will be 1000. So you always need to multiply the magnification of the objective lens with the eyepiece lens to get into the overall magnification of a compound microscope. Okay, so so is the resolution or resolving power. So that is the main key to all these different kinds of microscopes that we have, because there is always a resolution limit to each microscopes, and to overcome that we go from one microscope to another microscope. Okay, so in this part, in this slide, I will try to explain you what is resolution or the resolving power. So the resolution can be determined by the resolving power of your instrument. Okay, the overall resolution of your image can be means it can be means the idea can be drawn by looking into the resolving power of your instrument. So more the resolving power of the instrument, the higher resolution to it can go. Okay, so what is resolution? Resolution means to the distance to which you can actually separate two points which are very close to one another in space at separate entities as separate entities is known as resolution. Means there are two points in space which are very close to one another. So to what level you can actually tell that these 2 points are not a single point, they are two distinct points. So if you take the example of this image, so if an instrument with a low resolution power, it will visualize this as a single dot. Now if you increase the resolution a little bit, you find that no, this is not a single dot, there are two dots which are merging together. Now, as you increase the resolution more, you can actually differentiate these two dots distinctly. So the resolution with the resolving power with which you actually get this image is more as compared to the resolving power of the instrument with which you get this kind of image. Okay. So what you see is that the distance between these 2 points is increasing. So if the distance between these 2 points means if there are these small, very small distances, if you can visualize, means if one instrument can visualize a distance between 2 points around 10 angstrom, and another instrument can actually visualize two picometer range distance between two entities, then we will say that the instrument which can visualize these picometer range entities, they are actually giving you a higher resolution than the instrument which is giving you in angstrom level. Okay, so why I said you, I will tell you in a moment. 1 minute. Okay, so we are discussing about resolution. So the power of an objective able to separate distinctly two adjacent points is your resolution. And this resolution depends upon two things. One is the wavelength of the light that you are using for looking into that object, and, and the numerical aperture of the lens. Okay, so you remember that we talked about these different kinds of lenses here. So each of these lenses have a numerical aperture. So what is the numerical aperture? If you take the diameter of your lens and divide it by the focal length, so every lens will have a focal length. So diameter of the lens divided by your focal length gives you an idea about the numerical aperture of the lens. So it is actually giving you an idea. The more the numerical aperture of the lens, more amount of light can come through it, and it will help you to visualize your object more distinctly. Okay, so you can actually means visualize like that. Means if your numerical aperture is small, then less amount of light will be coming through it, and you will not be able to get a larger image, means or a clear image. You imagine like that you are in a dark room and you have two torches, one which has a wider dimension the wider dimension through which the light is coming, and another one it has a smaller dimension. So the light in which it has a wider dimension, it will be able to illuminate the object or that room more than those which has a smaller aperture. Okay, so that is the concept of numerical aperture and wavelength of the light used. For example, if you are using a red light means that it has a longer wavelength. As we discussed previously, if you remember, red lights have longer distance. What it will do is that since it has a longer wavelength, it will have more diffraction. Means if you want to travel that red light from one area to another, it will be diffracted by a larger amount, and this will, if the diffraction is more, then that can't be captured it means correctly through a detector. So this will lead to the lower resolution. So this is through logic. Means through logic you can actually tell that how this wavelength and numerical aperture is able to means give you the idea of resolution. But that has been proved mathematically as well. So that comes with your famous Abbe's equation. Okay, so if you see, the resolving power of a human eye is 0.25 millimeter. The light microscope can separate dots that are 0.25 micrometer apart, whereas the electron microscope can separate the dots that are zero point 0.5 nanometer apart. So why this is so? This light microscope can actually go to micrometer level, or the electron microscope can go to nanometer, to 2 nanometer level. Why that is so? Because there is this concept of this wavelength of your photons and the electrons. Since the wavelength of a photons are larger as compared with the electrons, it can only separate two dots in around micron level, in micrometer level. But the electrons have a much lower wavelength as compared to the photons, due to which you can actually separate two dots to nanometer level, means it will give you a more higher resolution as compared to the photons. Okay, so how you can connect all of these things mathematically, that is through Abbe's equation. So that is given as d is equals to zero point 0.5 lambda divided by n sine theta. Now, this n is the refractive index of your sample. Okay, so what is the refractive index of your sample? That is your n. And together, this n sine theta gives you the idea of the numerical aperture. What is d? D is the minimal distance to distinguish between two close objects. Means if you see that the minimum distance to distinguish between two close objects, so if this d value is small, that means that you can go to very small amount of distance, and you can actually visualize in that small amount of distance, you can visualize 2 points at distinct entities. That means if this d value is less, your resolving power is more, your resolution is more. So this is very, means some peoples are always doubtful about this. So you should clear it. D value more does not mean that your resolution is very more, is larger. Okay. So D value gives an idea about the minimal distance which you can distinguish between two close objects. So the smaller the D, higher is your resolution. Okay. So if you now consider that the smaller d means lambda, if it is large, if it is more smaller, then this d value will be more smaller. And since we know this d value is inversely proportional to the resolution, means if d value is smaller, your resolution will be more. Now we know that the lambda, or the wavelength of photon, is more as compared to the electron. Means for the photons, the d value is larger as compared to the electrons. which means the resolution of electrons is more as compared to your photon. Okay, so from here you see that what we were getting, these data, this can be actually related by this equation. Now n sin theta is the numerical aperture, and I mentioned you that more the numerical aperture, more is your resolution by a sheer simple logic. Now if you look into this Abbe's equation, you find that if n sin theta is more, d value is less, and d value is inversely proportional to resolving power, means your resolving power is more. So two things should be taken care of if you want to increase the resolution. One is you need to use a less wavelength or less smaller wavelength. You need to use a smaller wavelength, and you need to increase the numerical aperture. Okay, so if you are able to do that, then you will be able to get a higher resolution. Okay, so that you can actually get from this famous Abbes equation. Okay, now we are actually moving to the electron microscope. So since we now know the basics, why we have had to shift from the electron from the light microscope to the electron microscope, we now have the idea about the difference between the photons and the electrons. Okay, so how these photons and electrons means are behaving differently, and why you are using electron as a source, all these things are little bit clearer to us now. Okay, so now we are moving to electron microscope. So if you see the top image, we will first try to compare the light microscope with an electron microscope, because the basic idea in both of them is same. Only what is the major difference in between them is that in one case, for the light microscope, you are using more towards the photons, okay, or you are using light as a source. Whereas in electron microscopes, you are using an electron beam as your source. Okay, so there are different kinds of sources which through which you can produce these photons or light. Similar to that, there are different kinds of sources to produce this beam of electrons. So there are different electron guns which are present, which can actually provide you this beam of electrons. Now, as you see that once the light is coming and you want your specimen to actually means capture a condensed light, because this light, if it falls on this specimen, much of the light will be lost and it will not interact with your specimen. So you are condensing all the lights which are falling which should fall to your specimen through a condenser lens, and that will fall into a specimen that will move through your objective lens, and your objective lens will make sure that your light will fall into a detector or in a projection where you can actually visualize your, means this is your eyepiece lens where you can visualize your object specifically. Means these are the different lenses through which your light is getting condensed, and this condensation is very important to get a higher signal, otherwise much of the signal will be lost. So similar to that, people now wondered, okay, so we will use electron as a source. So for that we know that the wavelength of electron is lesser than photon, so we will get a higher resolution. But how to actually create this kind of lenses? Because if you want your electrons to get condensed through your glass, it will not happen. Okay, so electrons will not interact with the glass and get condensed just like your light. Okay, so how to, so then we will not be able to use these glass lenses for the electron microscope. Then how to produce these lenses? So now what we use is the property of an electron. So as you know that electrons, when moving through one place to another, it will produce electromagnetic waves. And these electromagnetic waves have an electric component as well as a magnetic component. Now people thought that if we can use, instead of these lenses, glass lenses, we will use certain electromagnetic lenses, where we will produce certain magnetic and electric field, due to which the electric magnetic field produced by these electrons will get a effect, means it will get repulsion, or somehow these electrons will now not be able to freely move when moving through these different magnetic electric fields of these electromagnets. Okay, that will actually allow us, if you actually change the voltages of these electromagnets, it will produce different magnetic fields and it will deflect the electrons in a different way. That will actually allow us to condense the electrons and fall into your sample. So more the electrons will be interacting with your sample, more amount of transmission, I would say, because here you are seeing that this is the electron beam is getting transmitted and falling into your specimen, and from there what it can happen here, two things can happen. Your detector can be in the downward direction or it can in a upward direction. You can get this scattered, the electron scattered information through it, as well as the electron transmitted through this sample. These two information you can get. So on the basis of this, your cryo-electron microscopes are also divided into two different types. Okay, so we will actually talk about it in the future, in the future slides. Okay, so as a nutshell, if you see the difference between a compound light microscope versus a scanning electron microscope, so the light microscopes cost is around this $150 to $15,000. Okay, but see, for electron microscope, it is very expensive. So cost wise, your electron microscopes are very expensive and they are more, means expertise is required to handle this electron microscope than the light microscopes. Okay, and the very fine tuning should be there so that your electron microscope should work, because if it these electrons it should move in the vacuum, because if these electrons interact with air, then many of your electrons will be interacting with your air and it will not be able to interact with your sample. So maintaining these electron microscopes is also very difficult and it is very expensive as compared to the light microscope. So what is the method of visualization? It, as I said, it uses visible light and a system of lenses to create and magnify the images. Whereas in electron microscope, you uses electrons to scan specimen to create and magnify images with the help of certain electromagnetic lenses. Okay, with the help of certain electromagnets, you are able to do that. Now, what is the magnification? So, magnification here for light microscope, it is around 1000 to 2000x at most. Whereas for electron microscope, it is very high, that is up to 50,000x. Why that is so? We just discussed about those resolution and everything. Okay, so in magnification wise also you get very high magnification in electron microscope as compared to light microscope. As we discussed, resolution is very low as compared to the electron microscope in light microscope. Image type that you get in light microscope, that is a 2D images you get. So there is a specimen and you get 2D images of your specimen through mostly your light microscope, were generally through your light microscope. Whereas in means there are exceptions, like there are different confocal microscopes which can actually give you 3D images. So there you actually use the idea of pinhole. But generally, you for a normal bright field or dark field microscope will give you 2D images. So since I talked about bright field and dark field microscopes, so there is a common confusion that bright field means their field is bright. Okay. Hello. 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 Am I audible? Means which part you were not being able to means get the means voice. You learned about this light microscope versus electron microscope. Okay. Okay, after the magnification part. Okay. So after the magnification part, that we discussed that means light microscope has a lower resolution because you don't get much detail, whereas in electron microscopes you have much higher resolution, you get a lot of detail. Then I said that in light microscope normally generally you get 2D images. But why I said generally? Because there is a light microscope, if you know the confocal microscopes. So these confocal microscopes can provide you three-dimensional images because of this pinhole concept. Okay, but generally, light microscopes like the bright field microscope or the dark field microscopes can give you 2D images. Okay. Whereas in electron microscope, you get three-dimensional images. Okay. Clear or detailed three-dimensional images you can get through an electron microscope. Now, 2D images means I said about bright field and dark field. So I was mentioning that there is a doubt always some people has that bright field microscope, it does not mean that your sample is bright. You can see the sample bright in a bright field microscope. On the name itself, you can actually clear your doubt. Bright field microscope means your field is bright, and in the bright field you are getting your images which are appearing darker. Whereas in dark field microscope, it's dark field microscope means your field or your background is dark there, whereas your images that you are getting, that those are brighter. So that is a dark field light microscope. Okay, so that confusion may happen for some people, so that's why I wanted to clear. Now, what are the advantage of light microscope? That better for observing living specimen. So why? So this is very good because you can visualize dividing cell movement, because there you are not damaging your sample. In light microscope, you will not damage that much your sample, whereas for electron microscope, it will be damaging your sample if you are not cryofreezing it. So there you get the idea of cryofreezing. Why you are freezing your sample? Because when you are heating your specimen with electrons, it will interact with the electronic levels of your with the atoms of your specimen, and it will excite some electrons that will actually give some radiative energies, like some heat energies will be produced, and also x-rays are produced as a byproduct of this electron microscopy. So normally when you see these electron microscopes like the here, where you can visualize that this electron microscope image, the cryo-electron microscope, this long tunnel, this is the tunnel through which your electrons come as a electron beam. But you see there are three coverings around this tunnel. What are the functions? One of them is the lead, because as you remember that to absorb the X-rays, you have this lead coatings when we talked about this in X-ray crystallography. So in cryo-EM, one of the byproduct of these electrons, when it is interacting with your sample, it will produce heat as well as some X-rays. And if that means the person operating that electron microscopes get means exposed to those x-rays, that can be harmful for him or her. That's why you have large lead coatings around these cryo-electron microscopes to protect you from those harmful x-rays. Okay, so normally if your samples are not cryo-freezed, then what will happen is that your sample will specimen will not be in a living state. So specimens are held or fixed in a place and unable to view living specimen, because much of your samples will be degraded. It will be dying through that heat. So that problem is being administered by this cryo-electron microscope, where you actually cryo freeze your sample. So you are not allowing this heat to hamper or to evaporate your sample, because this heat will try to evaporate your sample and you will not get the particular image of your sample as well. Okay. So now we are moving towards the different types of electron microscope. So as I discussed, so when I was discussing this electron microscope, I said that this detector can be in the bottom or in the above. Means two phenomena we can actually detect. One is scattering of these electrons or reflection of these electrons after interacting with your surface of the specimen. Another thing, how these electrons are getting or transmitted through this specimen, so that you also can detect. So from the name itself, so the electrons, when you are actually detecting the transmitted electrons, this falls under the transmission electron microscopy. Whereas this same, okay, scanning electron microscopy is interested in the electrons which are getting scattered or reflected by the specimen. Then you are collecting those electrons and detecting the surface. So scanning electron microscope can give you idea about three-dimensional images of your main samples you can get with the help of these electron microscopes, but it can give you, the scanning electron microscope can give you a three-dimensional image by, means how you can get this three-dimensional image, you can ask me, because I said you that in electron microscope you have vitreous layer of water in which your samples are embedded. Now you should get a 2D image out of it because you are scanning one surface. Okay, but these 2D images are being taken for various different layers of your water, and there is a process of image reconstruction. Means at different angles you will take these 2D images, and through computational tools, there are different tools through which you can actually reconstruct the 3D image from these different frames of 2D images. So that's why you get three-dimensional images. So in scanning electron microscope, you can get three-dimensional images, but since the electrons are not going inside your specimen and coming out, rather it is reflecting from the surface itself, you can only get the surface level or surface topography can be visualized with the scanning electron microscope. But for a transmission electron microscope, since here the beam of electrons are moving inside the specimen, and then through the specimen the transmitted light that you are getting, you are then visualizing that transmitted light. That's why you can actually, for transmission electron microscope, you can actually visualize the internal structure of, for example, a bacteria. If you have a bacterial cell, you imagine the bacterial cell as a cuboid, and there are, in different layers of that cell, there are different organelles or different things, different proteins or different mechanisms are happening. So that you can actually get with the help of a TEM. So the internal structure you can get with the help of TEM, whereas the surface information you can get with the help of the scanning electron microscope. But the mechanism is similar what we have seen for the normal electron microscope. Here, what you are doing is that you, the means mostly what we discussed for means the till now, the electron microscope, we discussed about this stem where you have an electron beam. It is the condenser is condensing that electron beam into your specimen, and in that space, through that specimen, some electrons is getting transmitted, and that is being collected by the objective lens and the projection lens. So sometimes your image is not in a, means virtual, or it can be, it's not in a real image. Okay, so the you get a virtual image. So to correct that virtual to real image, you get these projection lenses that will give you the amplified image of your sample. But since these electrons are interacting with your specimen and going through your specimen, this is transmitted through your specimen, you can get the information of each layer of your specimen or sample. So this is the general architecture of your TEM or transmission electron microscope. Scanning electron microscope, on the other hand, there is a scanning coil or a beam deflector which will actually deflect these beams and fall under this condenser. There are different multiple condenser lenses. So you want, because you want a surface topography, you want to scan each point, right? If you remember, we discussed when the types of microscope, we discussed that how you want to visualize your image. That is also you divide the microscopes. So in TEM, you are seeing the image as a whole. But in scanning electron microscope, you want to move from one point to one. You want to scan in XY direction. And then you want to reconstruct the image so that you get an image in the Z direction as well. You get a three-dimensional image. Okay. So in scanning electron microscope, what you do, you have this condensed electron beam, and you will move this electron beam in XY directions through your sample, and these electron beams, when get interacting with your sample, it will get reflected and fall under your detector, so that you can get this three-dimensional images of the surface of your sample. Okay. So this is the basic difference between the TEM and the scanning electron microscope. Now, on the basis of this, these cryo-EMs can be again divided into two types: transmission cryo-EM or scanning cryo-EM and scanning cryo-EM. So we are more interested towards this transmission cryo-EM because it gives the whole information, the information that you get in a transmission electron means cryo-EM, you will get to the subcellular locations, because you are actually getting information about all the layers. Okay. So this transmission cryo-EM can be further divided into single particle cryo-EM or cryo-electron tomography. Okay. Now, if you want to look into the general procedure, the total procedure of your means how these different sample preparations are happening, you can look into this. So the first and the foremost thing that one person should look into the while performing this structured means characterization everything, so that is on the basis of your means the preparation of your sample. If you have your sample, so the first criteria is to get your sample. So in X-ray crystallography, it was that you need to get a sample which is in higher concentration, and you need to produce a crystal of your sample. Whereas in NMR also, you need your sample to be in milligram quantities, and in NMR you wanted to label your samples with N15 or C13, which are NMR active, which have NMR active nuclei. For cryo-EM, it is not the case like this. In cryo-EM, you don't. That is the beauty of cryo-EM. You do not need to alter your system with the help of certain labeling, or you do not need to form crystals. All you need to do is you need to make some layers of samples. Means the foremost criteria of your cryo-EM is that you need to have highly homogeneous and pure sample. Homogeneous, by which I mean that if you have a pure sample also, means you have a single protein, but if that single protein is present at different conformations in space, then that is not homogeneous sample, that is a heterogeneous sample. So you want to visualize, since it will be reconstructing the 2D images to a 3D image. So this homogeneity of your sample is of utmost importance in the cryo. It should be homogeneous, it should be sample appears, it should be homogeneous. Okay. Then only you can actually proceed with cryo. So there are different procedures of this sample preparations of cryo means. Basically, if you means in a nutshell, what I can tell you, you can go in details and look into sir's slides, because sir discussed it in a very in depth. I am just giving you means the summary. So in cryo you have a grid. In the grid, you want to have your sample which is actually plunged in the grid with a in a very low temperature, means you have temperature of around one means 110 degrees Celsius, I think. So there we will learn about that, the process of vitrification. So you are where you are plunging your this sample in this grid in such a way that it will form vitreous layer of your solution like water, if your sample is in water, and you are cryo freezing it, so that in each layer you have homogeneous distribution of your sample. Okay, so in that vitreous layer of water, okay, then only you will be able to actually image your sample through this cryo. Now what is negative staining, out that we will be discussing in the next slide. Okay, now once you have negative stain your sample, in each step you want to first see whether after means just like in X-ray crystallography, we wanted to first see whether our protein crystals are formed or it is a salt crystal. Okay, then only we will be proceeding with the further steps. So just you negative stain your sample, you visualize it and try to reconstruct the sample and try to see whether your sample appears homogeneous or not. If your sample appears homogeneous, then you can actually go for the cryo-EM visualization. Okay. Now, then once you have visualized your sample, you need to actually also sometimes what you need to do is that sometimes you need to optimize your grid. Okay, to grid in which your samples will be present. So different kinds of grids are actually used. So you need to optimize it which kind of grid is actually useful, that will not hamper means the homogeneity of the sample, that will not be hampered by that kind of grid. You need to choose that kind of grid for your sample in which you will actually produce this sample with the help of different staining methods like this negative staining method. You actually need to do that. So that takes a lot of time. And then finally if everything is okay, means if sample is not aggregated or if it is not heterogeneous, means if it is heterogeneous in the grid you will see, and you need to again prepare your sample. You need to optimize your grid in such a way that in each layer the samples that you have they are homogeneous in nature. Then only your electron microscopy technique will work. Okay, finally if everything is okay, then you can go for the cryo EM data collection, where you will get this single particle reconstruction of your images. Okay, so this cryo-EM data collection you will get, and you will get the single particle means you will get the cryo-EM data. Now you have it's not like that a single virus or single I am taking the example of a virus because through the cryo-EM many virus particle structure can be visualized. So there is, if you imagine that if you have a virus solution, there will be not a single virus that will be imaged through this cryo-EM. So there will be individual virus particles or virion particles that will be imaged. So then you will have, finally when you will have an image, you will actually do a single particle reconstruction. You will average the whole the images of all the different single molecules that you have imaged, and you will get a final reconstruction of your image. So how these image analysis are done, and also what are the different terms you will actually come through like convolution, convection. So these things we will be discussing in the next class, where the various visualization techniques sir has introduced. So that I will be discussing in the next session. Okay, how this from the 2D image you are reconstructing into the 3D images. Okay, now in the sample preparation, so this is very important because this is the most important step I would say before proceeding, because other steps, there are many of the things, you have softwares through which you can analyze the image, but if your samples are not prepared well, then no means no question of getting a better three-dimensional image. So I will discuss with you here about the negative staining method, but there are other processes as well. So mostly negative staining methods are used in the cryo-EM means conventionally, but you can go through such lectures little bit and you try to see what are the different methods through which your sample preparations are done. If you have any doubt, you can actually ask me in the next class. So in a 2 hour session, it is very difficult to give the you each and every idea about the means whole idea. So you can actually go to search videos and look into it, but what is important I am telling you. So to perform single particle EM, thousands of identical particles are imaged and computationally averaged together. The ideal single particle specimen is thus homogeneous as possible. So here also you are getting the importance why your sample should be highly homogeneous, because as I discussed that there is not like a single particle image you take images of single particles of means the multiple single particles, and then you do a computationally averaging those images to get into your final image. Now you imagine if your samples are not homogeneous, then while averaging the structures, because the samples are not same, they are heterogeneous, maybe there can be some contamination or there can be some conformational changes due to which and sorry due to which the means the homogeneity of your sample is compromised. So then you will, during this computational averaging, it will give you a erroneous data, or it will not give you a image of higher resolution, because this all will add to the noise. Okay, so heterogeneity can be, how these heterogeneities can result. Heterogeneity can result from both conformational variation, as I mentioned, like for example, if your protein has large interlinker domains, like which do not have a particular domain, instead it has a lot of loops like region. So then for those regions you will actually have this means this heterogeneity of the sample you can look into the look there. Okay, so this how these heterogeneities are means then their samples will be conformationally heterogeneous, means their, because of these loop regions it can actually take multiple conformations in space, so due to which it will make the sample conformationally heterogeneous, or by compositional changes. For example, that's why I said you that you need to optimize the grid as well. So if there is, after you make the samples somehow interact with the grids, or the grids is actually changing the compositional the composition of the your sample is somehow changing by the effect of your grids, so that you also need to check. And also sometimes it's the effect of a binding partner, like the enzyme and the substrate. So if you have a substrate, then the enzymes will have different conformations like open or closed conformation, that can actually add to the heterogeneity of your sample. Okay, so what how the sample preparation is done. So you actually take the, means this is the process of negative staining I am telling you. So you have a sample, you take that sample in a grid. So it's for here we are taking a carbon support film, so they can be different for different grids are there. So and this is supported by these grid parts. Now what you do, you add heavy metal stain. Why heavy metal stain? Because, so what is negative staining? Normally, why it is known as negative staining? From the name itself, it is indicating something. So normally, when you are looking for some sample, you stain it with a particular dye, right? You stain it with a particular dye, and then you want to visualize it, so that dye will stain your sample and will help it to get visualized than the other samples in the media. For example, if you add ETBr, ethidium bromide, in a DNA sample, it will uptake that ETBr and will try to give the bright bands wherever this DNA are present. So that is an example of positive staining, means you are using a stain to visualize that object. What is negative staining is that by doing this staining, you are not visualizing your object directly. You are actually visualizing something else. You are visualizing something else, and due to the contrast of your sample with the background that you are visualizing, you are getting the images of your sample. So you can visualize it in like the dark field microscope. So where you have your dark field and in between you have certain bright spots. Okay, so that is the use of this negative staining. So here what we use is that we use heavy metal salts that can go and interact with, means that should not interact and may hamper the homogeneity of your sample. Instead, what it will do, it will surround your sample. Wherever your samples are there, it is surrounded by this heavy metal salt. And since these heavy metals have a lot of electrons, larger amount of electrons than those which are present in your sample, because in your sample it is made up of biological samples and made up of more carbon, oxygen, nitrogen. So amount of electrons which are present in those carbon, oxygen, nitrogen, that is much lesser as compared to that of the heavy metals. So these heavy metals will actually emitting out more amount of electrons as compared to this carbon, nitrogen and oxygen, due to which you will get dark spots for these heavy metals. And since these heavy metals are surrounding your specimen will be appearing as light. Like as you see here, this light, there will not be much electron density in your specimens, but in the background. As a result of that, you will get the image of your sample. So you put the heavy metal salt with your sample, and then you actually cryo freeze it at around 110 degrees Celsius, means 106 degrees Celsius, and means it's around this region that you should flash freeze your sample or you cryo freeze your sample, so that you get this vitreous layer of your samples. Okay, then you air dry it to actually means evaporate the overall means the extra amount of solvent, and only the heavy metals is surrounding your samples like this. And when you image this in the microscope, it will look like this. Okay, now this is means this samples, how it is actually cryofished. It is cryofished by putting it into liquid ethane, which is around minus 60 degrees Celsius. Okay, and it will actually freeze your sample like these layers, means in each layers of water, where the outside layer is freezed, whereas in the inside there is liquid water. Okay, so in this way you can actually get the image of your means you can prepare your samples. Now someone can ask that why we need to flash freeze. What will happen if we are not flash freezing it? Then there is the possibility of formation of ice crystals. Now, if ice crystals form in your sample, then what will happen is that these ice crystals will also means transmit these electrons, and that will actually add to the noise of your sample, and it will not be able to give you high resolution images. So one is that, and another part is that you want to freeze your sample, you want to protect it from the heat generated by electron microscope, but also you want to provide an in vivo like condition where you can actually get the dynamics, or you can actually visualize the macromolecules in more in vivo like environment. So that's why we are actually flash freezing or cryo freezing it. Okay. So what is the process of cryo immobilization? That is known as vitrification. So this cryo immobilization preserves that aqueous environment, which is also ameliorating radiation damage. Imaging of cryogenically immobilized samples by EM is known as cryo-EM. Okay. So this is the definition of cryo-EM. By just when we started cryo-EM, we have went through it. Then the sample must be frozen extremely rapidly at a rate of 106 degree Celsius per second. So 100 degrees Celsius per second, you should reduce the temperature per second, so that the water in and the surrounding of the specimen is fixed in a vitreous state. Hydrostate means a thin film of water where outside is frozen but inside the water is in the liquid form. If freezing occurs too slowly or the specimen is subsequently warmed above 137 degrees Celsius, the temperature at which water devitrifies, crystalline ice is formed. Okay, so that I just discussed with you in the last slide. Now finally we are moving to the detectors. Okay, so they means once you have actually prepared your sample, you have done the electron beams are hitting your sample, and it is going to where? It is going to a detector. So there are different kinds of detectors like charge coupled detectors or like direct electron detectors. They are also known as these direct electron detectors. Okay, so through which you can actually get the images of your cryo-electron microscope. So now sometimes what happens is that these detectors can also add to certain noise in the image, and that you don't want. So ideally, so for each detector, there is this detective quantum efficiency, that is this DQU, which is the signal to noise ratio of your output signal. Means once this detector is receiving one signal, that is your input signal, and the signal that is going out from the detector to your computer, to which you are actually working on to the image reconstruction. So that is the output signal. So the signal to the noise ratio of your output signal square divided by signal to noise ratio of your input signal square, that should be ideally equal to 1, but that is never the case. So you need to choose your detectors accordingly where this DQ value is means near to 1. Okay, so this is about little bit about the detectors. Now, very important thing. One thing is about the protein data bank. So once you have made your three-dimensional images of your sample, and you have passed through all the refinement tests and everything, and you have a higher image around resolution image around 2 to 3 angstrom. So one of the limit of this cryo-EM is that you can't go to that much resolution that you get for X-ray crystallography. Means in cryo-EM you always get a resolution around 3 angstrom. So if you get with cryo-EM an image around 2 angstrom, so these are now possible by different kinds of grids. Okay, so you change the types of grids and you get into different kinds of, means now the resolutions are also getting better for this cryo-EM. Okay, so that I am not discussing, but there are many modifications of these grids through which you can actually get very high resolution images of cryo-EM as well. But once it has passed, then you need to submit it to a database. So one of the database is this protein data bank. So this protein data bank is a database for the three-dimensional structural data of the biological molecules. So it comprises the data of all X-ray crystallography, NMR spectroscopy, or any cryo-electron microscopy structures, three-dimensional structures in higher resolutions. So structural biologists submit their structures in this protein data bank. So now one thing is that this PDB ID means every protein data bank, every structure that you submit in protein data bank, that has an identifier or a PDB ID associated with it. Now this PDB ID is normally, the convention is that it should start with a number always. The first, so there will be four alphanumeric values, where the first alphanumeric value is always a number or a numeric or a number or an integer between 0 to 9. Okay. Then the other three letters are normally, generally, they are alphabets. But sometimes, means not the second one, the third and the fourth one can be also sometimes, okay, can be also numbers. But normally, the convention, the general convention is that this first alphanumeric value should always be a number followed by some alphabets. The three are the alphabets or the letters which can take from A to Z. And the convention is like that. The more recent it is, like if it is one ABC and another one is two ABC, then the 2ABC is a much recent structure submitted to PDB than the 1ABC. So this 0, 1, 2, 3, 4, this also tells you about the generation, the to which generation of to which time period this structure is submitted. If it is 1ABC, 2ABC, so they are first generation, second generation submission of your PDB. The more recent structures, it will have much higher number here. But then again, someone can ask that it will have some limit at a time. So now this PDB is also means if you go and visit, they are trying to means extend this ID to some other chain IDs and other all IDs will be now inserted. So that means in future they can do. Sometimes what happens, this ABC, these all are should be in block letters, but sometimes they can be also represented as in small alphabets like normally this is done for this O and 0. So this O is not can be if it is given in small small alphabet, not in a block letter. Okay, so there you can actually easily distinguish 0 with O. Otherwise it is quite confusing. O and 0, they can be in block letters. Sometimes this I and L. So this L and capital I and small L can be confusing. Okay, so if you have L and I together, so it is conventional that you should use either block letters or small letters, but mostly you should always use the block letters for this alphabets. Okay, so this is about the PDB ID. Now the data typically obtained, they are all from X-ray NMR and cryo-electron microscopy that can be submitted in PDB. And on 2021 they celebrated their 50th anniversary. And the first 3D electron microscopy is the bacteriorhodopsin. So that we discussed in the first slide of your cryo-EM. Okay. So then there is specialized another data resource from where you can actually get only the electron microscopy images. Okay. So the cryo-electron microscopy enables the determination of 3D structures of macromolecular complexes and cells from 2 to 100 angstrom resolution EM data resource. And it's so this to that level you can get the 2 to 100 angstrom resolution, you can get the image. And this EM data resource is the unified global portal for one-stop deposition and retrieval of 3D EM density maps. So the electron density maps, the three-dimensional electron density maps, they are actually these atomic models and all the metadata associated with your electron microscopy images that can be actually found in this EM data resource. Okay, so this is all what I wanted to discuss with you. So first, let us discuss the assignment questions. Then, if we have time, we can go through these practice questions. Otherwise, you can look by yourself in home. I will supply you the slides, so you can look into it. So first, Shiny, if you what doubt you have in the assignment, can you tell? First, let me clear that. Then we can discuss all the solution. Hello. Okay. So is it this question? The maximum resolution of transmission. This one, right? Okay. So in the transmission electron microscope. So let me go to the solution of it first. So the transmission electron microscope has means 2.5 angstrom. It has the resolution, right? Hello. Um, am So zero point 0.5 angstrom is the maximum resolution of your transmission electron microscope. As this is a means slide that I have taken from source means material only. So zero point 0.5 angstrom means 10 angstrom equals to 1 nanometer, right? So zero point 0.5 angstrom means then 0.5 divided by 10. right? So point 5. Oh, no, no, no. It's around point 0.5 angstrom. It's point. It's given in such a slide. Okay. In the transmission electron microscope, if you go, so the sir mentioned that it is below point 0.5 angstrom, to which you can actually go. The resolution, the maximum resolution of this electron microscope. Okay. So it is from here. I actually they made the question. So zero point 0.5 angstrom means zero point 0.5 divided by 10 means 0.05 nanometer. Now it is clear? Okay. So other than this, you have any doubt? Okay. So so let me discuss the all the questions one by one, and then we can actually discuss little bit today. If you have any doubt, we can discuss about that. So the first question is, what is the general representation of PDB ID where X is an integer and B, C and D are any alphabet? So the first thing what you should always think is that the PDB ID always start with an integer. So it should be X, which will be first. Okay. So option A and option C has that. X is first, but then C also there are two X. Okay, B and C you can actually neglect because the first alphanumeric value should be a number. So in among this A and C, I said you the second one should be always an alphabet. Sometimes it may happen that the third and fourth has some number, but general convention is that the third, fourth will have an alphabet. Okay, so as given by the convention. Okay, so that's why the answer to the in the second position here there was also a number, so it is also wrong. So the correct answer to this question is option A, right? Then, what is the final detector of cryo-electron microscopy? So, the final detector of a cryo-electron microscopy is a CCD camera, which is known as the charged coupled device. And from this slide, I will discuss that it is a charged coupled device. So, the answer to this question is C, charged coupled device. 3D image can be visualized by which of the following technique? Three-dimensional images can be visualized by which technique? Okay, so 3D images, as I mentioned, that dark field microscope is a light microscope. It will give you 2D image. Compound microscope will again give you 2D image. Magnifying glass will give you a 2D image, and it will not give you an image. It will only try to give you a magnified image, but that is in a 2D. That will not give you a 3D image. Okay, so only the electron microscopes I said that they can give you 3D image. So scanning electron microscope option is there. So the option here is A. The correct option is A. The PDB provides information on protein structures determined by, as I said, all X-ray crystallography, NMRs, electron microscopy, all these structures are deposited in PDB. So the answer to this question is D. The whole virus can be best viewed in. So from this, we mentioned that in electron microscopes, you can actually get the whole 3D image of your virus because that is in the nanometer range, the virus size, nanometer angstrom range, the virus size. So that's why, and also this same, you can ask that same and cryo-electron microscope, but the whole virus, it is telling you the whole virus. So in same, you can only get the topography information, the layer, the outside layer information. So in cryo-electron microscope, you can get the whole information about the virus. So the answer to this question is C. Then another five questions, that is, which of the following term used for rapid freezing of the sample at a rate of 106 degrees Celsius per second so that the water in and surrounding specimen is fixed in a vitreous state? So that is the vitrification process that we discussed in detail. Okay, today. So the answer to this question is from this option or question also you can get vitreous state. So that from there it is coming vitrification. Okay. The internal structure of a bacteria can be best viewed in which of the following technique? So the internal structure. So I said the layer by layer structure can be visualized by TEM, whereas the outer topography structure can be visualized by SEM. So layer by layer, the internal structure of a bacteria that can be visualized by your TEM, that is your transmission electron microscopy, which is the first 3D microscopy structure released in the PDV. So 3D electron microscopy structure is of your bacteria rhodopsin that we discussed. Okay, so the answer is option D. Maximum resolution of your transmission electron microscope, that is zero point 0.5 angstrom. 10 angstrom equals to 1 nanometer. So zero point 0.5 angstrom means zero point 0.5 divided by 10, that is 0.05 nanometer. So the correct answer to this question is option C. Then the power of an objective able to separate distinctly two adjacent points is known as. So this we discussed in details. So that power of an objective to separate any two points in space as distinct entities is known as the resolving power. Okay, and the phenomena to which means it is happening that you can distinctly identify two points in space, that is given by the resolution. Okay, so the answer to this question is option B. Now, if Shani, you have any doubt, you can ask me. Okay, if you have any doubt, you don't have any doubt. So you were able to get the all the things right that I discussed today. Okay, so we have some time. So since we started little bit late today, so you can actually solve this practice question and tell me the answers if you can, because these are very easy if you follow today's discussion, right? So then I will give you the answers. You can just note down the means questions and try to answer. Okay, and you can write your. Okay, so instead of just telling, you can write the answer in the chat box. Then we can actually go through the. I can look into your answer and also discuss the answer for others. Okay, so you can just write your answers in the chat box. Okay, once you are done, you can just tell me that yes, I am done. Then we can move for other 5 questions. Okay, so I have 10 questions. Okay, you are done. Hmm. Okay, so so let us discuss the first 5 questions answer. So I will just note down your answers first and try to check. So you said 1A, 2B, 3A, 4B, 5C. Okay, so one you have given very easy question. So it is in the millimeter scale that we can actually visualize to our human eye. So the option of the first answer is A. So good. And then the second question, what is the final detector of cryo-electron microscopy? So this is the assignment question. So I took it from previous year. So see, this got repeated in your assignment. So it's the answer is C. So I hope you have given the correct answer. 1 minute. So you have given 2P. What is B? No, so it is charged couple. Okay, okay, okay. So yeah, yeah. So I know because you have given this answer in the assignment, so it's correct. It should be 2C. Yeah, it's fine. Then in the third question is surface topography can be visualized by which of the following technique? So the surface topography on the surface, only the surface you can view, that is through the same, that is the scanning electron microscope. So what answer you have given? It is A. Good. So then in the what is the full form of PDB? I hope you have given the correct answer. So it is Protein Data Bank. So these are very simple questions, and these are only asked in your questions in your papers in the exam as well as in an assignment. So no, nothing to worry about. You can just enjoy this course, learn something new. That is the motto I think of sir as well as my 2 hours of discussion always. That if you have know if you know something, you should add some knowledge from this session. Okay, marks and this all will come. Okay, so this is very easy, so the option is B, and I think you have given yes. So good. And the final one is the whole virus can be viewed. So this is again the repetition of your answer. So this is cryo-electron assignment, that is cryo-electron microscope. So the answer is correct answer is C. Option is C. Yeah, good. So you have given all the correct answers. So now let us go to next 5 questions, and after that we will discuss the solution, and that will be end today's session. Okay. So again, the first question you can ignore. So that is again the same. Okay. So much of the questions you can only solve the question number 8 and 9. Okay, and this is because I discussed today, and 10, 8, 9, 10 you can answer, because all other questions are actually means the you can write those answers, but this is already in your assignment and we discussed today. But yeah, still you can write the answers. These are very easy, I think. So yeah, it's fine. But these kind of questions will be asked. So it's good that you practice little bit. Sometimes another question that I have not seen and I have not given here, but I will tell you that this kind of question can be asked. You can look into the Abbe's equation, because from there any numerical can come, means from the Abbe's equation. So you can actually look into it. You can actually try to memorize the formula, okay, of Abbe's equation and the concept behind it. If some question is not numerical, they can ask you some qualitative question from that. Okay, so that yeah, yeah. So yeah, good. Then you can always ask me any doubt. Means if you have any question from your course, that is that I have not provided here, but that is related to your means the topic that I am discussing here. So you can just come up with those question, and I will be happy to solve it if you have any doubt. Okay. So any numerical problem if you get through a base equation, okay, you can just come. If you have any doubt, you can come and ask me, sir. I don't know. Means how to means I am facing difficulty. Can you solve? So that will be good. That will be practice for you as well as those who are watching it later in the YouTube. Okay. So you can, these are important points. And then in means resolving power, there can be many questions that can be asked by means increasing the numerical aperture, or the means sometimes we use oil, means what is the use of oil in the microscope. Okay, so all those things, those are all related to this numerical aperture and everything. So you can, these are some important topics you can actually look into. And from cryo, some technical parts, I don't think sir will give too much mathematics, because this is difficult for those who are working in cryo. Okay, so the all the structural means analysis part, that I don't think, only some information oriented question he can ask, and some important concepts you should take. Okay, so that is important. Like what is cryo? If someone asks you, can you be able to explain that guy in very simple language? So if that is there, then you have learned about the technique correctly. Okay. Okay, so I think you have already given your answer. So let us discuss. Yeah, so 6D, 7C, I will just check it, but I think it's correct because this is only we have discussed. So yeah, so sixth question. 6D, yeah, vitrification. 7 is C, the internal structure of bacteria, because we are moving, looking into the interior of a bacteria. So the option C is the correct. And then you have this, which is the first 3D electron microscopy structure revealed. So that is bacteriorhodopsin. So 8D, good. And then again, so how many years? It's 50 years. So it's a very boring question, but this type of questions are asked, so it's fine. 9C. So yeah, fine. Then the power of an objective able to separate distinctly two adjacent points. So that is the resolving power. So I think this all you have given correctly. So yeah, I what I will do is that apart from these questions, next class I will try to bring some numerical questions, if some means some tough numerical questions. Okay, so that can actually help you to get some idea about some numerical questions. So these are very easy. So if you don't have any doubts, Ali, we can leave today. And in the next class, I will be talking about the analysis part little bit, and let us